Okay, you guys, I'm giving this another try. But this time, I'm not going to read the book, I'm going to read my lecture notes. Okay? So, this lecture is about the visual brain, perception, and action. And there is a lot of graph, but I'm not going to show it to you because I don't know if I can. Lols, copyright issues and stuff. Anyways, this is just a lecture write up, like a summarized version of the lecture. It made sense to me at the time. I don't know if it will make sense to me now. Anyways, let's get into it. So, let's compare the differences between hearing and vision. So, in the physical medium, what are we detecting? So, in hearing, we're detecting frequency and amplitude of vibration, and the sound traveling through the air. Hold on. I handwritten this on my hand and my hand. What was it? My penmanship is not the best. Okay, so in physical medium, in hearing we are detecting sound, while in vision we're detecting light. And light is basically frequency and amplitude of vibrations traveling through the air. Oh geez. While light is intensity and wavelength of light reflected from object. So in hearing, the transduction, I think like where the hearing takes place, is in the ear, specifically the organ of corti in the cochlea. While for vision, it's in the eye, specifically photoreceptors of the retina. So that's kind of clear. So, but how does it take place? So for hearing, it's pressure goes to the basilar membrane and then to the hair cells, and something about action potential. I don't know what that is, but I do remember in the last video, in the last one, I was talking about how sound move, I think, and you get the pressure from the outer ear canals, and then it goes into the inner ear, or is it the middle ear, where the basilar membrane is like there, and then basilar membrane is like the top and the tympanic is the bottom, I think. Oh my gosh. My final is tomorrow, you guys. I don't know if I can do this. Why did I pick brain science? Anyways, okay. I should go for a cheaper one, I swear. Anyways, and then like between the two membranes, there's the hair cells that does something, and then something about action potential. I don't know if we covered this. I don't think we did, so I don't know what this is actually. Anyways, so in vision, the how does vision take place? So you have light that goes into the photoreceptors, and then turns into photopigments, and then there's this word action potential again. Okay, so what features are being represented? So for hearing, you have pitch, you have loudness, you have, what the hell did I write? Beat fan. Hold on. What is that? Oh my god. Vision? Anyway, auditory scene analysis, grouping, whatever that is. Okay, anyways, I think you just need to remember pitch, loudness, grouping. It's always safe to remember like three things. But I have to actually. Anyways, never mind. Ooh, let's not panic. Okay. Mm-hmm. And in vision, you get contrast, color, and edges. There's also grouping, shapes, surfaces, whatever. So, the first part, the visual brain. So, in previous lectures, we have learned about the physiology of the eye and the brain and how these link to perception of color and depth. Importantly, the retinogeniculate striate pathway shows how the visual system becomes increasingly organized. This organization tells us about the features being represented as well as the functional specialization of different areas. Let's consider the perception of edges and lines. In the retina, the center surround organization of ganglion cell receptive fields means that they are sensitive to relative differences in luminance. Thus, more firing will occur in locations where there is a change in light. These sorts of receptive fields are also found in the lateral geniculate nucleus, where inputs from the retina are segregated according to their origin in magno and parvocellular neurons. In area V1, simple cells are extremely sensitive to lines of a particular orientation. This area of visual cortex is highly organized, reflecting the visual field retinopathy and the eye of origin ocular dominance. Elsewhere in V1, complex cells have larger receptive fields and are sensitive to moving edges. So selective encoding starts in the retina with centers around receptive fields, and then it goes through the LGN or the lateral geniculate. Wait, hold on. I don't remember what LGN is. I don't know, you guys. Okay, hold on. LTN is something like geniculate. I don't remember. I know it was hard to pronounce. That's what I remember. And then it goes through V1. Whatever that means. And the number of fibers in the optic nerve is only about 10% the number of photoreceptors. So it only passed about 10% of the info. Whatever that means. And then organization in visual cortex is demonstrated by the properties of simple and complex cells. Yeah, so I didn't cover vision because I'm hoping it doesn't come out. I mean, it obviously does, but I'm not gonna answer it. So you have for IMTK at a master's level. So my exam is basically to write 2 essays in 2 hours about 2 questions. And the questions are always like, you have one about vision, you have one about hearing, you have one about perception, and then you have one about like multisensory stuff. So yeah. Anyway. So basically, I figured if I master like two of the basic things, I'm okay. So I thought, why not go for hearing and then perception, right? Because that was the easiest to learn at that point of time. But like, apparently, I don't retain anything. Anyway. This description shows that as we move through the visual system, neurons become more and more selective and thus they represent more and more complex features. Okay. Anyway, for the visual brain, the visual system is extracting information rather than just representing every pixel. The retina encodes relative brightness and contrast difference. The LGN organizes MP cells and information from the two eyes. Cells in V1 respond to edges, motion, and corners. Okay, so part 2, the 2 visual streams. Oh, okay, so I like this one a lot. I like the stuff about the brain. So this one talks about ventral and dorsal streams. So what basically they do. I like this one a lot. This was my favorite part of the lecture. Let's hope I remember stuff. Okay, so a different but influential approach to perception is based on the function of vision. It is clear that one of the main functions of vision is to guide the motor system in performing actions. Research in cognitive neuroscience emphasizes that after V1, information from the eyes is processed in 2 streams. So the ventral stream, which travels through the inferior temporal lobe, is thought to be primarily involved in recognizing objects. So it analyzes the what in the scene. So inferior temporal lobe, if I'm not mistaken, hold on. Ventral stream is the one that goes around your head. Yeah, it's at the sides, while dorsal is like at the top. Okay. I'll put up a picture of a brain, I guess, for you to see. Anyway, the ventral stream, which travels through, the dorsal stream proceeds from V1 into the parietal lobe. This pathway has been hypothesized to represent where objects are, and in particular, those attributes are important for perceiving and acting on moving objects. Oh my gosh. Okay, so I think this is the answer to one of the past questions about perception and action. And like, you get information from your eyes, and then what's important is how your brain processes it, right? So then it talks about this experiment in 1982, which is kind of horrific. You'll get what I mean when I get into it, because okay, so I'm just gonna read it and then I'm gonna talk about it. So, the original evidence for two visual streams was from experiments in non-human primates by Ungerleider and Mishkin in 1982. So, in these experiments, monkeys had to learn to retrieve food by either recognizing the container or by learning its spatial position relative to a landmark. Animals with lesions in the inferior temporal cortex disrupting the ventral stream could no longer perform recognition tasks but they were able to learn the spatial problem. Animals with parietal lesions, hold on, my notes are all over the place. Disrupting the dorsal stream showed the opposite pattern, intact recognition but impaired spatial learning. Sorry guys, I'm not good at reading, I'm good at talking, but not reading anyway. Thus, these experiments provide evidence for a dissociation in the brain between the representations required for the two tasks. So, what this means, what they did basically is that they took monkeys, right? They have two groups of monkeys. So, first, one group they like tasked them to learn how to recognize objects and how far it is from them, right? I think they do this for both. So both groups do that. You know, they learn the task and oh, this is a banana and this is how far it is. So then they took out parts of their brains. So one group had their ventral stream, which if you were listening, was sorry. I'm talking too much. I love this topic. I don't know how to say it. So ventral stream, which is involved with object recognition. So it tells you if this is a banana or if this is a cup and stuff like that. So one group of monkeys got that taken out from their brains. They actually did brain surgery on monkey. Okay, and then the other group had the dorsal stream, which is where the object is, so like spatial location and stuff. So after that surgery, uh, they they did the task again with the monkeys, and like the group with the parietal stream, sorry, with the ventral stream messed up, had the cannot recognize the object while the but can tell how far they are, while the ones with the dorsal stream messed up, they know what object that is but their sense of depth is strong. But I also learned from like I don't know if it's in this lecture or like in the next lecture, that even though their eyes can't perceive location right, their body still has it right. Like they can still reach for an object and they can still reach it at the right, how you say, at the right distance, you know, even though they don't see it right. So I think they did there as some scientists also did this experiment with humans without the brain surgery. I don't remember what experiment that is. I have to look back at my notes because the book is not helping anyone. Yeah, so I think they just use like some kind special goggles, I think, just to, or like to mess with the perception of distance. So you know, like some glasses have that effect on you, like when you look into a pool, your feet looks like shorter than normal or longer than normal. You know how that it's like all the light and stuff, blah blah blah, but uh your hands or your feet can still reach to the object right, you know, even though you see like this doesn't feel right. So yeah, so that might have different control in parts of the brain, right? So with the ventral and dorsal stream specific, so for sorry, for ventral stream it's the um parietal lobe, was it, or is it temporal? Hold on, blah blah blah. Yeah, temporal cortex is ventral, and the parietal lobe is dorsal. Okay, so that's it. And then, okay, to continue, Milner and Goodale, I don't know, 1995, extended this theory to humans and refined the distinction, arguing that the dorsal stream is really a how system devoted to using visual input in the control of action. This vision for action system represents the immediate state of the world in an egocentric frame of reference so that we can act on things at the current time. The ventral vision for perception system is characterized by longer lasting view independent representations, which allow us to consciously recognize items. So both streams use both object and spatial info, but the purpose of the dorsal stream is to support action. Like what I said, you need a vision for perception, like you need to see the spatial location and stuff, but you can kind of still reach for it. Yeah, stuff like that. So so part 3 is dissociating vision for action. Several behavioral experiments have demonstrated action without perception, which supports the idea that there are two separate systems. Well, yeah, this is what I just said. For example, some visual illusions that affect conscious recognition, like the dorsal stream, have a reduced or different effect when participants are required to perform an action. In the Ebbinghaus size contrast illusion, a circle is seen as larger or smaller depending on the surrounding shapes. Okay, so Ebbinghaus illusion is basically like you have two same circles, but like one is surrounded by circles that are smaller than it, and the other is surrounded by circles that are bigger than it. So it makes the illusion that the circle at the center is not of the same size when it actually is. So that's the Ebbinghaus illusion. There's a lot of illusion you have to remember for like seeing and hearing, or like advanced cognitive psychology one. So however, when picking up a circle in the same illusion, people could match their hand shape without being fooled. Agliotti, Goodale, and D'Souza, 1995. So this and other examples imply that action can occur without an accurate conscious perception. So visual illusions can fool our perception, but it cannot really fool our motor system, because the action system is hold on, is right, but even when perception is not. So there's also a rotating mass illusion. You can Google this online. I think YouTube has it. So one like either when how like mass have like that thing protruding, and then another one like a hollow part to put your face in, but so when you rotate it, like the hollow part still looks like a face, like it's concave. I'm not sure. Yeah, so yeah, you have to like see it to believe it. Anyway, so in neuropsychology, some patients with visual agnosia have brain damage to the ventral system, which is consistent with the role of ventral system in conscious recognition. One of the most studied patients, DF, has severe problems recognizing even simple shapes, but the vision for action approach suggests that she might still be able to act efficiently based on the dorsal pathway. Milner et al. 1991 said that. So basically, ventral stream, what they found so far, supports conscious awareness and are susceptible to visual illusions, while dorsal streams are not so easily fooled. But Ebbinghaus illusion shows that while it's not easily fooled, it can still be fooled. It's just like if you talk about vision for action, it could be sorry, ventral is the vision for ventral is the vision for perception. The whatever. Okay, anyway. I'm almost done, don't worry. So the last one is anyways, not yet over. Anyway. For example, DF can post a card into a slot. So DF is like the patient's name, but you can't like say the person's name. So this is for anonymity. For example, DF can post a card into a slot even when unable to consciously perceive the orientation. A possible counterpart. Okay. So agnosia is basically like you can't recognize object. And prosopagnosia is you can't recognize face, but you can recognize objects. So anyway, so a possible counterpart to agnosia is optic ataxia. Ataxia, I don't know. This disorder is associated with damage to the parietal lobe in the dorsal stream. So optic ataxia appears to be due to. Sorry, I can't pronounce this. Ataxia. I am having trouble with anything with X in the name. Optic ataxia appears to be due to how visual information is transformed into a representation for action. So basically, you have trouble reaching for things, because you can't really like perceive how far the location is. So, uh, patients with ataxia perform better, but with a delay. If you're asked to think consciously. Despite the convincing evidence for perception-action distinction in the brain, the cognitive processes in each system are unlikely to take place in isolation. For example, we often need to recognize an object in order to act appropriately. Crane and Profit, 2001. This will require information from both visual streams interacting. In Cream and Profit's experiment, interfering with the semantic what system still had an effect on vision and for action. I don't know what that means. I don't know what this is. Okay, hold on. When you load the ventral stream, it affects the action task. Thus, it shows that the two are not completely separate. So, I think what it means is that I'm not sure what it means. Never mind. Wow, my stomach hurts. I shouldn't drink chai tea anymore. It always makes me have weird stuff. Anyway. Last part, biological motion. Biological motion describes the pattern of human movement which are easily recognized by observers. This has been well demonstrated by Johansen, 1975, who used point light walkers as a highly controlled representation of movement. These stimuli consist of a few spots of light filmed by placing markers on the joints of human actors. Despite being very simple, Participants are remarkably good at inferring what the actor is doing, as well as their gender, emotion, and other traits. Perceiving biological motion often involves attributing animacy, agency, and intentions to moving objects. In a classic study, Hyder and Simmel, 1944, showed animations of squares and circles moving around a screen. Observers spontaneously saw the movements as interactions between animate agents, even describing the personality of the moving shapes. It is clear that perceiving animacy this way goes beyond merely analyzing the visual shapes in the scene. So what they did was what they're saying basically is that even without like context or anything, if you just like show a bunch of shapes and then move them around, people can still like make a story of what happened. I think there is a I don't remember the name of I don't remember the name of the experiment specifically. I think they basically show like there are squares and circles and triangles and like the triangles was close to the circle, but then the triangle moved away and then comes the square and then stuff happens and then like people can kind of make a story from that. So that's basically what it says. So that's about it. We talk about the visual brain, stuff about the eye and how like the brain gets how light goes from the retina to LGN to V1, and then from V1 you get the two visual streams, which is ventral and dorsal. So ventral is for the what system, which is the object recognition, while dorsal is for the where for spatial location. So ventral, which is in the temporal cortex, temporal lobe, yeah, is a vision for perception system, while dorsal, which is in the parietal lobe, is for the vision for action system. So, and then we talked a little bit about agnosia, um, and how you don't have to um your like action, like your vision may be full but your action may not be. And then you also have like agnosia, but you don't we'll talk more about this in like the next topic, because it talks more about like agnosia and prosopagnosia, which is about the same as agnosia, but we'll also talk about how do you perceive objects and scenes. So, and then lastly, we'll talk about how we can easily describe patterns of human motion or human movement from like just a bunch of simple shapes shapes moving, stuff like that. So I actually enjoyed this. I think I learned a lot. I just need to remember the experiments. Those are always like the empirical evidence that I need to remember. But I am feeling more confident. This is actually a video where I actually learned things. See this? I should have read this from the start. Damn it! What the hell? Anyways, okay. I hope you learned something. Thank you for listening. You can always search for stuff that I said immediately because I am too lazy to put them in. Um, but whenever I have time or whenever I feel like it, which I most probably won't, I'll put in all the stuff in the links, sorry, in the description, stuff like that. Okay, so I'm just gonna, yeah. So this one will just have a picture of the brain because I'm lazy. So, goodbye.